Welcome to this course, Introduction to Complex Biological Systems. So, this will be the last lecture of week 5. In this lecture, I am going to discuss cancer, right. Cancer is a collection of diseases. So, all of these diseases are concerned with cells, different types of cells. So, you can think of that there are so many different cell types. So, there are so many different cancer types. Some of the most predominant cancers are listed here on the y-axis, and the x-axis tells you the number of people who are infected by this disease per year, right. And this data is taken from United States, right. So, you can see that some diseases, some cancers are more prevalent and some cancers are less prevalent. And so, in this case what you see is in green shows the number of people in thousands who are affected each year, who are diagnosed with this particular cancer each year, and in blue shows the number of death. So, it turns out that cancer has become one of the major killers, right. So, this is in the context of United States, and here is some data for India. So, in the year 2022, 14,61,000 people were diagnosed with different form of cancer. Again, considering the population that we have, this number is underestimated. So, there are many people most probably from rural areas who are not getting diagnosed. So, 1 in 9 people are likely to develop cancer in his or her lifetime in our country. It turns out that lung cancer is most prevalent in males and breast cancer is most prevalent in females. The incidence of cancer cases is estimated to increase by 12.8% in 2025 as compared to 2020. So, that is a big jump, right. So, with more aging population and with a large population, this has become a real issue for our country. So, one thing that I would like to point out is that lung cancer, which is most prevalent in males, and it has a very good correlation with smoking. So, in this plot what you can see, so this is years, so this is time, and these are the number of cigarettes smoked per year, right, by people. The red line shows you global cigarette consumption, right. So, how the global cigarette consumption has increased over the year. So, now with all governments have put in a lot of effort in educating people about the problems of cancer. So, this number you will see is slowly coming down. The green line shows the global lung cancer deaths caused by smoking. So, you can see there is a direct correlation. So, of course, it takes some time for cancer to develop. Therefore, this green line lags this red line, but they look very similar, right. So, it is following exactly the same trend as that of cigarette smoking, and this blue line shows lung cancer that is unrelated to tobacco, and that is very less. So, this is something that we will see, that there are causes of cancer which are sporadic, but then there are also causes of cancer which are directly correlated with our lifestyle. Okay. So, this slide shows you some of the basic events that happen during the formation of cancer. So, the cells that you see here, these are called epithelial cells, right. So, these are the ones that are on the top. Now, let us say one of these cells gets mutated. Now, this mutation can happen during cell division. So, in the last lecture we have seen that stem cells are there. There are cells that die. So, stem cells have to divide to replenish the to replace the dead cells, right. So, when that happens, when DNA replication is happening, there can be errors. So, that can lead to mutation. Also, errors can come from some other agents. So, errors can come from the internal process of DNA replication. Errors can come from external agents. We will see some of these agents in the later slides. So, let us say for some reason mutation is caused. Now, that mutation happens in some critical protein so that the cell can divide more rapidly than normal cell. So, this is called hyperplasia, right. So, here these cells look very similar to normal cells, but they can divide faster, right. And while this happens, again since you are getting so many cell divisions, so many DNA replications, again more errors can occur. So, some cells will gain additional mutations in some critical proteins or genes, which will lead to this increased growth potential, right. So, now you see this extra cells which are different from these cells, right, and those cells will divide rapidly, and what we get is a tumor. So, at this stage this will be a benign tumor because this tumor is localized in a particular site. So at this point you can actually cut it out. So if this is diagnosed in time, a good surgeon can cut it out so that they can remove this whole mass, right? So no more cancer cells. However, if detection is not done in time, there is a probability that these cancer cells will gain some more function due to more accumulating mutations, and they can break this tissue and they can start moving, right? And once they get into the bloodstream, they can go to different parts of the body, and these cells can accumulate in different places and start growing as tumors. So now tumor will spread throughout the body of the person, right? So this is invasive cancer, and the last stage is metastasis, which is very difficult to treat. So if you are interested in cancer biology, then these are some of the terms that you will come across, and it's a good idea to get yourself familiarized with these terms, right? So the first one I have already talked about, it's hyperplasia. So it is the increase in the number of cells in an organ or tissue. So increase in number than what normally it should be, right? However, the architecture of the cells are normal. So it looks very similar to the normal cells. Then we saw benign tumor. So it's not aggressive, it does not damage the local tissue, and it does not spread. So it remains localized in a particular place, and this can be removed surgically and the patient will be fine, right? Malignant tumor, which is now cancer, it is aggressive, it can damage the local tissue and it can spread. So we already saw that in the last slide. And metastasis is when these cells get into the bloodstream, get transported to different parts of the person and start growing as tumor at a distant site, right? So this is metastasis. Now it turns out that there are different types of cancer, and depending on the cell type, they get different names. For example, you will hear of carcinomas. So carcinomas are cancer of epithelial cells. So for example, breast cancer, lung cancer, pancreas cancer, right? So these are all carcinomas. Adenoma is a benign tumor formed in epithelial cell. So if it is localized in a particular place, and if it's a benign tumor, it will be called a adenoma. Sarcomas are cancer of connective tissues, right? For example, in muscle cells, when that happens, it's called leiomyosarcoma, right? Cancer of blood cells is called leukemia. The same blood cells, the most likely the white blood cells, so when they accumulate in lymph organs such as thymus or spleen, then that will be called lymphoma, right? So these are all cancers, but depending on the cell type and the location, they are given different names. Okay, so just a reminder about the cell cycle. We have already seen this, that a cell, it decides to divide, right? So there is cell growth and chromosomal replication that happens in the S phase. Then in the metaphase, chromosome segregation happens, and then we get the cell division. So two daughter cells are formed, right? And we have seen in case of stem cells that it can be symmetric division or asymmetric division, right? Now many things can go wrong. So when the chromosome is replicated, mutations might occur because errors in DNA replication, right? When chromosome segregation is happening, errors might happen if the mitotic machinery for some reason does not work properly in that particular cell. So you might actually, instead of getting 2 chromosomes here and 2 chromosomes here, you might get 3 chromosomes here and 1 chromosome here, right? So those will also create problems. So repeating pattern of cell growth, including chromosomal duplication, and cell division, including chromosomal segregation, can lead to cancer, right? Okay. So again, we have seen this, that there are several checkpoints, right? So this is your typical cell cycle: G1 phase, S phase, G2 phase, and M phase. And there are checkpoints which monitor these phases and ensures that no problem has occurred. But when there are mutations in proteins which are involved in these checkpoints, then the probability of getting cancer increases. So what happens when this cell cycle regulation goes wrong? Then uncontrolled proliferation or cell division happens, which can lead to the formation of tumor. So cell division without checking for DNA damage, right? So again, when the DNA is getting replicated, DNA damage can happen, mutations can happen. So there are proteins, there are enzymes which are called DNA repair enzymes, which identify these mutations, which identify this damage and correct them, right? But if those corrections are not made, it can lead to cancer, right? So DNA replication errors are not corrected. DNA damage can happen during replication. DNA damage can also happen due to some chemicals which are called external mutagens, right? So again, if those mutations are not corrected, they can lead to cancer. But again, remember that not just one mutation is enough. There has to be multiple mutations in critical proteins or critical genes. When that happens, then the chances of cancer increases, right? So here are some numbers. So error rate of DNA polymerase is 1 in 10 to the power 5, right? So now our genome is 6 billion base pair long. So you can see how many mutations will happen. It turns out that in one round of replication, there will be 1,20,000 errors, right? So that's a huge number of error. However, I told you that there are DNA repair mechanisms, right? So there are proteins which will detect these errors. They will detect this mismatch in the DNA that has happened. They will come and correct it, right? So all of this reduces this from 1 in 10 to the power 5 to 1 in 10 to the power 9, which means that 3 to 6 mutations will happen during one round of replication. And then again, by chance, most of those mutations will happen in places which are not critical, right? Again, mutations might happen in a genome, in a gene, but the mutation might not be such a mutation that it changes the characteristic of the amino acid, right? So when I talk about evolution, I will talk about this in more details. So you will see that in that case, what happens is that the mutation will not have any effect, right, on the structure or function of the protein. But in some cases, this mutation can create problems. So the chances of that are less, but considering the number of cell division that happens, if you add that, then you will see that the chances of getting a problematic mutation increases, right? So, but in most cases, all of those are taken care of by mechanisms that are present in the cell, like DNA repair. But when mutation happens in those enzymes which are responsible for DNA repair, then we have a problem, right? So during replication, DNA polymerase has proofreading activity, so it will reduce this error. After replication, a well-designed DNA repair machinery repairs the damage. So that also reduces this number. So it ultimately reduces it to 1 in 10 to the power 9. But if DNA damage is beyond repair, then the cell dies by initiating apoptosis. So there is something that is also there. So I have not talked about this, and you will see this in the later lectures, that if the repair cannot be performed, if the DNA damage is beyond repair, then it will trigger certain events in the cell which will result in the death of that cell, which is called apoptosis, right? So this is regulated cell death, right? Now, when this DNA repair or apoptosis, when both these fail, then we have a chance of cancer, right? So there are many checks and balances present in the cell which prevent from cancer in occurring. Okay. So how DNA damage happens? So this is DNA. There are several factors. I talked about replication errors. You can also get cancer from ionizing radiation. Mutations can also happen from UV exposure, right? So we will see that UV light, it can cause certain reactions, so it can result in DNA damage or mutations. Now this is something that is very interesting. So in the next week's lecture, I will talk about vitamins, and you will see that there is a particular vitamin, vitamin D, which is synthesized only when you get UV light, right? So UV light triggers a particular reaction which results in the formation of vitamin D, and vitamin D is very important for us, right? So you need certain exposure to sunlight, but overexposure can cause unwanted mutations in the DNA which can lead to cancer, right? So there is always a balance, and this is something that you will see in biology, that there is always a balance. If the balance is upset, then problems happen. There are also chemicals which are mutagenic. So mutagenic chemicals can cause mutations in the DNA. There are reactive oxygen species which happen due to certain reactions that happen in our body. They can also create mutations, and of course there are proteins and enzymes which deal with this type of reactive oxygen species. Cigarette smoking, so that's lifestyle, right? So when someone smokes cigarette, then tobacco itself has certain mutagenic chemicals which can cause DNA mutation, and we have already seen example of the data of cigarette smoking, how it is related to lung cancer. So these mutations can cause these problems. So they can create pauses at the checkpoint. So mutations happens, so the cell cycle pauses at the checkpoint, and it can try to fix it, right? If that does not happen, there is a problem. If there are mutations, DNA repair program kicks in, it will remove that mutation and correct the DNA. So if that happens, then the cell will proceed through the cell cycle and everything is fine. If that does not work, then the cell will trigger this programmed cell death or apoptosis. Okay. So I talked about mutagen. This is one example. This is called benzopyrene, benzoapyrene, and it's a promutagen. So what is a mutagen? Mutagen is a chemical which will cause mutation in the DNA. So how do you check that whether a chemical is a mutagen or not? There is a test called Ames test. So one can take these chemicals and put it in bacteria. So there are certain ways you will do this experiment, and then you check for mutations in the bacteria, right? So if mutations happen in the bacteria because of these chemicals, you say that chemical is a mutagen. Now, benzoapyrene is a promutagen because if you do this Ames test with benzoapyrene, you will see there are no mutations, right? But people have seen that this is present in tobacco and it causes cancer, right? So what happens? It turns out that this molecule itself is not a mutagen. However, when it is inside our body tries to flush out this type of molecules. So it goes to the liver, and the liver will do certain reactions so that it can be easily flushed out. One of those reactions are these oxidation reactions, right? So this type of epoxides are formed, and it turns out that these epoxides are cancerous. So these are the actual mutagen. So if you add these epoxides in bacteria, you can see mutation happening, right? So that is why this molecule is called a promutagen, that initially it is not a mutagen, but when it enters our body, it goes through the liver, it gets oxidized into these type of molecules into these epoxides, which are mutagens, right? So this type of compound can be found in tobacco smoke, coal tar, in some food types, right? And especially in grilled meat. So if you take any meat which is grilled or little burnt, then in those burnt portions you will get this type of molecules. So based on what we are discussing now, that it seems like cancer, a major factor in cancer is mutation, right? So in that sense, you can consider cancer as a genetic disease, and there are many evidences of that. So this is how chromosomes look like in a cancer cell, right? So if you take a cancer cell and if you look at all the chromosomes, if you paint them with different colors using fluorescent dyes, you will see something like this. Now there are several abnormalities in these chromosomes. So if you think about our chromosomes, we have 23 pairs, right? So 1 to 22, and then the last one is the sex chromosome, so X and Y. And we have 2 copies of each chromosome, one coming from each parent, right? But if you see here, there are 4 copies of chromosome 1, there are 6 copies of chromosome 2, 3 copies of chromosome 3, right? So the chromosome number is completely random, right? It's not maintaining these 2 copies for each chromosome. This is something that is called aneuploidy, so abnormal number of chromosomes, and this happens in, you will see this in many cancer cells, right? So it means that when the cell is dividing, mitosis happens, and somehow the mitotic machinery is not working properly, so that the chromosome numbers are increasing, right? And this can create problem because so suppose there is a particular protein, you need only 2 copies of that protein, but if you have more copies of that protein, because there are more chromosomes, then there are more copies of the gene, so it will produce more proteins, right? So the protein dosage increases, and that can upset the balance. So this is one problem. Another problem that you will see if you look at this chromosome number 20, so there are many copies. Now if you just look at these, there are 2 different colors. So blue, which is actual chromosome 20, and there is also orange, which is most likely coming from chromosome 5, right? And again in chromosome 5, you see there is orange and there is blue, which is coming from chromosome 20, right. So there is a problem here, right, and this type of chromosomes are called translocations. So this process is translocation. So a chromosome abnormally, so these are chromosomal abnormality where a chromosome breaks and a part of it reattaches to a different chromosome. Okay. So you can imagine that there are two chromosomes. These are two different chromosomes. This chromosome breaks, the top part attaches here and this bottom part attaches here. So now you have a primary chromosome where the top part is from one chromosome and the bottom part is from a different chromosome, and then of course the reverse will be there in the other chromosome, right. Now proteins which are at this junction region, they will also get snapped off. So if there is a gene here in chromosome 5 and if there is a gene here in chromosome 20, those genes, part of those proteins will get swapped, right. And in many cases this type of translocations cause abnormal signaling events and lead to cancer. So here is sort of a summary of what I am discussing so far, that this is a normal cell, right. It will accumulate mutation due to different events. So it takes in one mutation which might result in hyperplasia, so the cell is growing more, and then it gains some more mutations which can most probably damage the DNA repair mechanism. It can gain some more mutation which can damage the programmed cell death or apoptosis. It can gain some more mutations, right, and things like that. So these mutations will accumulate to finally turn it into a cancerous cell. So this whole process can take years, right, and it turns out that there are 3 to 20 critical mutations which are needed. The least is 3 and it can be as many as 20 mutations in critical genes, okay, not any normal random places, right. They have to be in certain critical genes, and we will see what type of genes are those, and that can lead to cancer. So what are these types of mutations? It can be a point mutation, just one nucleotide changes to another nucleotide which can change the characteristic of the amino acid, which can change the function of the protein. It can be deletion, so a part of the gene is deleted, or it can be insertion, some other genomic part is inserted into this protein, into this gene, so that new amino acids are encoded. It can be frame shift, so maybe some one nucleotide is inserted so that the whole frame is shifted, right. It can also be gene duplication, so a particular gene, let's say it helps in the stem cells to divide, right. Now that particular gene is duplicated due to errors in replication. So instead of one copy, you have maybe 3 or 4 or 10 copies, right. So then the number of protein that will be produced will be more, and that can create problem, right. Translocations, as we saw in the previous slide, where different parts of the chromosomes get chopped up and attached to other chromosomes, and that can result in chimeric proteins. So chimeric proteins means there is a protein where one part of the protein comes from one gene and another part of the protein comes from another gene, right, and that can have a mixed function which is not there in any of the normal proteins. So here is a more detailed description of a mutation. So what do I mean by mutation? So let's say this is the original sequence, right, and you can see that these nucleotides remain the same. However, this T gets changed to C, right. Now if this T gets changed to C, if this is my codon which is CTG, that will get changed to CCG, right. So it can code for a completely different amino acid, right. In many cases it will code for the same amino acid because of codon degeneracy, right, but in some cases it can actually code for a different amino acid. And if that different amino acid is very different in characteristic, for example, let's say a hydrophobic residue gets converted to a charged residue, it can change the structure of the protein and the function of the protein, right. So a mutation is an alternation in the nucleotide sequence of the genome of an organism, and this mutation can result in an altered gene product, which is the protein. Spontaneous mutations occur due to replication error, and induced mutations are caused by mutagens. So we saw example of a mutagen, right. So what is a mutagen? It's a physical or chemical agent that changes the DNA sequence. So it can cause this type of mutations, right. So it can be UV X-ray radiation, it can be chemicals like benzoypyrene, which can come from tobacco chewing or tobacco smoking, right. So, when these mutations causes cancer, it will be called a carcinogen. Now, we have seen that there are two types of cell division, one is mitosis, one is meiosis, right. So, somatic cells, they undergo mitosis. So, in this case, if mutation happens, then that can cause cancer, but it will be in the same person, right. But if this mutation happens during meiosis in the germline cells like the sperm or the ovum, in that case, it will be passed on to the next generation. So, in this case, mutation will be transmitted to the progeny, that is the next generation. Okay. So, what are the hallmarks of cancer cells? There are these four major hallmarks. The first one is uncontrolled mitotic division, right. So, uncontrolled cell division, which is called tumorogenesis. The second one is evasion of cell death signals. So, it avoids apoptosis, right, which is immortalization. So, these cells will keep on dividing. Now, since the tumor is formed, it will need nutrition, right. So, it will need the formation of blood vessels. So, these cells, they will induce formation of blood vessels so that they can get nutrients and oxygen and all these other materials to divide faster, which is called angiogenesis. And finally, they will break open the tissue, they will damage the tissue and they will invade into other places, which is metastasis. So, normal cells will not do that. So, they stop growing once they have reached a certain stage, right. So, they will not grow on top of each other, right, but the cancer cells will do that. So, they can invade tissue, they can move from one place to another place. So, the first one is development of tumor. We have already seen this, that there is a mutation that results in hyperplasia, these cells are growing, then more mutations accumulate, right, and then the tumor grows, okay. But still this is localized, this is present in one place. Second one we saw is angiogenesis. So, they will induce formation of these blood vessels, right. So, you have a tumor which is growing here. Now, there was this blood vessel, but since the tumor is growing, it needs more nutrient. So, it will induce the formation of these blood vessels. Third one is immortalization. So, let us say this is a normal cell. So, it is a stem cell, let us say it divides, and while that division is happening, there is some form of DNA damage, right. So, in that case, it will induce this DNA cell repair mechanism. So, one of the proteins that is involved in this is named P53. We will see P53 is it's called also the guardian of the genome. So, what it will do is it will first arrest the cell cycle, it will stop the cell cycle, it will recruit the DNA damage repair proteins. So, the DNA damage can be repaired, and once that happens, it will allow the cell to proceed through the cell division. So, daughter cells are formed. If it cannot repair the damage is such that it cannot be repaired, then it will recruit the proteins which induce apoptosis, right. So, it will start the programmed cell death and result in killing of the cell. So, this is what happens in a normal cell. This is happening every day in your body, right. But during cancer, this normal process does not happen. So, let us say DNA damage happens because of intrinsic or extrinsic signal. Let us say there is mutation in P53 or for some reason P53 is not recruited, right. So, then there will be no cell cycle arrest, right. So, and also no recruitment of this DNA damage repair mechanism. So, there will be uncontrolled cell division and that can result in tumor. And finally, there is metastasis, which means that this tumor grows and then it damages this tissue so that it breaks open and gets into the bloodstream. And then these cells travel, they go to some other place and then they start growing a new tumor there. So, here the tumor spreads throughout the organism. Okay. So, what causes cancer? We saw that gene mutations causes cancer. Now, in normal cells, let us say these are stem cells, and we have seen examples that our body requires formation of new cells, like we have seen in case of red blood cell, we have seen in case of the gut cells, right, epithelial cells. So, you need formation of new cells, but this formation of new cells is highly regulated. So, when the new cells are needed, there are signals which will say, okay, go divide. So, they will start dividing. When the required number of cells are produced, then there are stop signals and the cell division will stop, right. However, in case of cancer, this go signal is hyperactive, right. So, they will keep on dividing, and this stop signal is also damaged, which means that they are not going to stop. So, they will keep on proliferating, they will keep on dividing. Now, the proteins which are involved in this step, which tell a cell that okay, now it's you have to divide, there are more requirement. This type of proteins are called proto-oncogenes, right. So, their normal function is to signal the division of a cell, right, and it turns out that there are 300 such proteins. When there are mutations in these proteins, they become oncogenes. So, they can lead to cancer. So, in cancer, there will be mutations in some of these proteins. Proteins which give this stop signal, they are called tumor suppressor genes, right. Now, if there are mutations in some of these tumor suppressor genes, then in those cells, of course, the cell division will not stop. There is no stop signal, and it turns out that there are almost 200 such genes. So, in total, there are 500 genes which are the critical genes, and mutations in some of these genes can lead to cancer, right. So, I told you that there has to be some critical mutations. So, those critical mutations have to occur in these type of genes. So, what are proto-oncogenes? Their normal function is to regulate cell growth, to regulate cell division, and to regulate cellular communication. So, cells talk to each other through their extracellular matrix, and they can tell that okay, there is some gap here, more cells are needed. So, they will signal the stem cells that okay, we need some more cells, so please divide, right. So, all of these things are regulated by these proteins. However, if there is a function, if there is a mutation in them, so that they themselves become hyperactive, right. So, there is no signal that is telling them that okay, now tell the cell to divide, or now don't tell the cell to divide, right. So, if that signal is gone and they are always telling the cell to divide, in that case that is gain of function, right, and that results in uncontrolled cell division, and these type of mutations are dominant mutations. For example, if you think about this particular gene, so there are two copies of this gene, one from each parent. Now, one is a normal copy, the other one is the mutated copy. If there is mutation in the mutated copy, then that mutation is enough to overcome the function of the normal protein or the normal gene, right. So, it will be a dominant mutation. The other type of cells that we proteins or genes that we saw are the tumor suppressor genes. They are involved in cell cycle regulation, DNA repair, and cell death, which is apoptosis, right. So, if mutation happens in this, then the stop signal is gone, right, and that will again lead to uncontrolled cell division. Now, these type of mutations are recessive mutations, right. So, it means that again you have two copies, one copy is corrupted, but the other copy is still there. So, it will still give the stop signal and the cell division will stop, right. So, mutations have to happen in both the copies, right. So, that is why this is a recessive mutation. Okay. So, I talked about p53, which is a tumor suppressor gene, right. So, in normal cell, division does not depend on p53 protein, because cell division is happening, there is no issue, right, everything is working fine. However, if there is a DNA damage that happens, that can happen because of the replication process itself, that can happen due to some chemical agent or UV radiation or some other radiation, right. So, in that case p53 levels will rise, and the first thing they will do is stop the cell cycle. So, arrest the cell cycle, bring in the DNA damage repair proteins. If all of these things work, then of course go through cell division. If that does not work, then it will induce cell death. If DNA damage happens and p53 is absent, right. So, for some reason there is mutation in p53 or p53 is not activated because of the proteins that recruit p53, then of course none of these things will happen. No cell cycle arrest, no DNA repair, no apoptosis, the cell will continue to divide, and that can lead to tumor formation and eventually cancer. So, p53 is called the guardian of the genome because it protects the genome, right. It ensures that the correct DNA is present and the cell division happens correctly. So, there are all these types of cancer, and now over the past 40 years all sorts of therapy have been developed. So, I talked about surgery, there is also radiation therapy. So, you use radiation to kill these cancer cells. There is chemotherapy, so you use medicine to target the cancer cells and kill them. We will talk about immunotherapy when we talk about immune system. There are targeted therapies. So, we saw some of that in the last lecture when I talked about stem cells, and then of course, there is stem cell therapy. So, I talked about these two in the last lecture a little bit in the last slide. So, these are the books. You can refer to these books for this course. Thank you.